Okay, welcome everybody. Thank you for joining us for the July Sci Cafe. I'm Anita Grove with the Apalachicola National Estuarine Research Reserve, and we host this monthly virtual series generally the fourth Thursday of the month. The series highlights research conducted in and around Apalachicola Bay. Will be probably about an hour in length. Our speaker will save time for questions, so we have a chat function. If you'd like to type your questions in there as you think of them, I'll read them off later and have Reuben answer them. So today we have Reuben Wilson. He's with the USDA-NRCS-Soil and Plant Science Division. And the title of our talk is Shellfish, Sulfides, and Soil Carbon, Coastal Zone Soil Survey of Apalachicola Bay. So since 1899, this National Cooperative Soil Survey has worked to develop a nationwide partnership of federal, regional, state, and local agencies with the goal to investigate, inventory, and document and interpret and publish information about soils. So Reuben Wilson is, like I mentioned, with the Plant Science Division, and he's going to talk about some of their initial observations over in East Bay where they've been sampling soils. So without further ado, Reuben, I'll let you have the stage. Thank you so much.
Yeah, thank you, Anita, for that introduction and having the opportunity to present and speak about some of our work that's been going on in the area. As Anita said, my name's Reuben Wilson. I'm a soil scientist with the USDA-NRCS, so the U.S. Department of Agriculture and Natural Resources Conservation Service, and the subdivision I'm part of is the Soil and Plant Science Division. So we're responsible for inventorying the nation's soil and plant resources around the country. And so, as Anita stated, today I'll be talking about some of our initial work on the Coastal Zone Soil Survey that began last fall in Apalachicola Bay. So kind of what I'm going to talk about today is, first I want to give a brief overview of like what soil survey is. Kind of, you know, it's my whole world, but it can be a little bit of a nuanced science that folks might not be familiar with, some of the resources that are available because of soil survey and how we really go about conducting it. Then I'm going to take it a step further, talk about the formation of Coastal Zone Soil Survey, how it came to be, why it's important, and why it's currently being conducted. And then I'm going to talk more so about the Apalachicola Bay project, its genesis, its progress, what we're seeing, what we're doing. And then finally, I'm going to go into some of the initial results. I was telling Anita earlier that we received lab data back, some lab data, not all of the lab data, about a week ago, and I've kind of compressed what I can here to speak about that, as well as show some of the photos of the soils and kind of explain some of the geomorphological processes that are going on there. So what is soil survey? Soil survey, in short, is a systematic analysis of landscape and landform relationships. And so what a landform is it is a smaller feature that occurs on a larger landscape. And so you can kind of visualize this, that a tidal marsh would be a landform, or a floodplain would be a landform, or a marine terrace would be a landform on a larger landscape, and that landscape would be like a coastal plain. So our landform would be that tidal marsh on the larger landscape, that would be that coastal plain. And so the idea is that we can subsection these areas of the larger landscape into these smaller landforms based on their repeating topographical, hydrologic, or vegetative features. So we understand, even though these landforms or these sections of tidal marshes aren't next to each other, they are on this same larger landscape of a coastal plain, and they exhibit these similar topographical, hydrologic, and vegetative features, and likely the soils on those landforms that exhibit these repeating features behave the same or form the same, have been subject to the same soil forming factors. And so what we do in soil survey is we conduct analyses of the soils on these representative landforms. And so we collect physical and chemical properties. I'll go more into that a little later in the talk, and develop a range of characteristics. So we understand if we have 20 tidal marsh sites, let's say, or 20,000 acres of tidal marsh in an area, if we go and inventory 10 or 15 points within those tidal marshes across those 20,000 acres, we can see based on our chemical and physical analysis and the range of characteristics that we've developed that allows us to infer what that soil is probably going to look like on an average tidal marsh in the larger landscape of that coastal plain. And so what this does is it allows us to create a model for a large area without extensive observations. You know, as much as I would love to transect every tidal marsh that exists across the larger landscape or in Apalachicola Bay, I have to identify certain ones that I believe are representative based on the factors that are occurring on that landform on the larger landscape to understand that the average tidal marsh is going to have soils that look like this and are within this range. And so what we do when we use this landform and landscape model and develop our representative soils is we get a map. And so this is what we refer to in soil survey as a block diagram. So what a block diagram is it shows a larger landscape. So this would be your larger landscape, if you can see my mouse cursor kind of going around here. Your larger landscape, and then, your larger landscape here, and then these kind of marked out areas with these names on them would be your land forms on that larger landscape. So using this soil survey approach or using soil survey allows us to understand that if we're on the flatter part of this landscape, further away from these high dunes or ridges or knolls, that this is probably what the soil is going to look like. And so you see that there's the name Mayaca right there, and that's if you want to think about it this way, when we classify these families all the way down to our soil taxonomic family classes, they're attributed with a local name so we understand, or it gives us insight as to where that soil was originally documented, inventoried, and understood to be occurring. And so the soil survey approach allows us to make this map of the landforms, the standard soil that usually occurs on those landforms across a larger landscape. So it's using that about understanding these similar forming factors and getting our representative soil inventory of that area, and then being able to attribute that to landforms across the larger landscape. So why are these maps important? You know, a lot of people are like, well, why are you going out and digging holes in the ground? Why is it important? What's it used for? You know, ultimately, when you develop these soils and these ranges of characteristics with these soils, what are you getting from them? And so what happens is when we generate these soil maps, we populate the characteristics, we understand all the important stuff is in our database so that we can develop interpretations for land use. So what you're seeing, those images on the right, are actually from the published soil survey in East Point or in the Apalachicola area there in Franklin County. And with these soil maps, it allows us to develop these interpretations. And some of the interpretations that we have for our soil maps are for dwellings with basements, shortleaf pine suitability, playgrounds, paths, trails. There's a whole host of different interpretations that we've developed based on certain soil characteristics, so that when somebody goes into our interface, which I'll get into in a minute, such as Web Soil Survey, and they want to develop a report to say, hey, where in East Point would be the best place to have a dwelling with a basement, you know, if anybody even has a basement in East Point, you can see here that this red indicates that that is poor suitability for a dwelling with a basement based on our soil survey map. This yellow would indicate some limitations, and then green would indicate there's no limitations. Likewise, when we move down to this next image here, the shortleaf pine suitability, you know, you can see it's fairly suitable for shortleaf pine production based on the interpretations in the soils that we have mapped there. So, you know, we're generating these soil maps, but we're also generating interpretations to help people understand what the soils are good for without expecting them to be a soil scientist and understand all the jargon that we use. So where can you access these maps? So all of these are publicly accessible through Web Soil Survey. And what you can do in Web Soil Survey is it allows you to choose an area of interest anywhere in the continental anywhere in the United States and territories as well, as long as there's soil data published there and publicly available. You can choose an area of an interest, generate a report, and you can access these different soil interpretations. You can select the ones you're interested in, what you might want to be doing, or maybe you're just curious, and it allows you to access this information. Also in Web Soil Survey, it's a geospatial information. So say you're working in GIS and you want to be able to pull in the soils layer for your area of interest, what you're curious about, what you're operating on in GIS, you can do that through Web Soil Survey as well. We have a geospatial data gateway where you can download all of the soils data in there, as well as also the interpretations. So it's a really good public resource to understand why we do soil survey and how you can use soil survey to help you interpret the land and what you're wanting to do. So there's also an app for it. Personally, I really enjoy the app. You can download it on your phone. There's also a desktop version, and it's called SoilWeb. I think it's a little more user friendly, or at least, if you're just curious like I am, as a soil scientist, wherever I go, I have SoilWeb downloaded on my phone. Whenever I'm in an area and I'm interested in what the soil might look like, I can hit get soil data, and it'll generate a colored render of the mapped soil, as well as some of the characteristics and soil suitability ratings for it. So another way to access the soils data that we have is through SoilWeb, which is more user-friendly, more fast-paced, but it doesn't necessarily give you all the information that Web Soil Survey would give you. We also have Soil Data Explorer, and that you can get the available lab data. So if we have sampled a soil to a certain depth, all of that is deposited into Soil Data Mart, or our Lab Data Mart, our access interface, where it's all public facing data that if you are interested in what the soils looked like in Franklin County for a certain area, if you can go through and use this tool, and you can see if there is soil data for that area, and if it is, you're welcome to download it and open it and use it as you need it. So that was a brief overview of what soil survey is. So it's the inventory of the soil resources on similar landforms that reoccur across the larger landscape. So the idea that all of these smaller landforms have similar pressures or features, hydrologic, vegetative, relief, topographical, and that they have similar soils on these landforms across the larger landscape. So now that we've went through and talked about kind of the basics of what soil survey is, I'm going to talk about really what coastal zone soil survey is and how it came to be. So as Anita mentioned in the opening blurb there about this talk, soil survey actually began in 1899. And that's when Congress authorized funds for the USDA to investigate and map soils to enhance with understanding the nation's soil resources, but also to help enhance agricultural production. The first soil survey was actually conducted in Cecil County, Maryland, for a tobacco farm. And so really the history of soil survey for the first 100 or so years of it really focused on agricultural productivity. Where is the best place to go corn? Where's the best place to go cotton? Where is it too wet to properly grow it? And so it was a really agronomically focused product. And so over time, because the areas, coastal wetlands, subaqueous areas were considered non-productive for agricultural uses, they really got left behind in this initial inventory of soil resources. And as you can see here in this image, this is from near Sapelo Island, Georgia. We talked about that soil rendering on that past slide. You can see here, this soil rendering just says no data. So in this instance, we have a data gap because this area wasn't considered agriculturally viable. And so resources weren't put forward to properly inventory the soils that occurred in these tidal marshes in these areas. And so this left a group, including people like the National Estuarine Research Reserve and others in and around Apalachicola that utilize water resources and these wetlands. It left them with a data gap of not understanding, what does a soil look like there? What could it be good for? Of course, we're not looking at agricultural production for it, but maybe we're looking at subaquatic vegetation restoration. Maybe we're looking at the best places for oysters, for example. And so we really had this data gap for this suite of land use managers, as public taxpayers that weren't being directly benefited by how they needed to be served through the soil survey. So coastal zone soil survey in itself really emerged in the 1990s. So in 1993, a soil scientist named George Demas from Maryland really pioneered this idea that, in subaqueous soils as well as these tidal marsh soils, just because they don't have the typical vegetation of these upland soils, they're still undergoing pedogenesis or soil formation, and therefore are supporting vegetation. So based on the congressional mandate put forward as far back as 1899, that it was our duty to inventory these soils just as well and make sure we're serving all of our customers across the United States, whether they be growing corn in Iowa, or if they're looking at the best habitat that can be grown or what kind of habitat can be supported for an endangered species in a tidal marsh or maybe in a subaqueous environment. So coastal zone soil survey really started gaining steam around 2010. That's when the first coastal zone soil survey was published in Rhode Island, and it has increased exponentially since there, because in 2024 we completed our largest project, which was the Long Island Sound on the Connecticut side, where we published or updated around 422,000 acres of soil survey. And so that's a photo of George Demas on the top there, and that bottom image is what some of the updated soil maps look like up in Connecticut after the completion of that portion of Long Island Sound. So now we've talked about soil survey and the landform landscape relationship, kind of what is coastal zone soil survey, how it came to be, and now we're going to go more into the Apalachicola Bay project proper and talk about it. So the background of the project: in the fall of 2023, the Apalachicola National Estuarine Research Reserve formally requested a coastal zone soil survey from the USDA. And so their priorities were really inventorying the coastal and subaqueous soils to assist with research, as well as understanding restoration practices, provide insight to accelerate oyster production recovery, and also with their surface elevation tables, kind of tying that to soil data and maybe understanding based on these surface elevations that might be gaining or losing, if we can help understand that and attribute that to different soil characteristics and property. So in the spring of 2024, after moving through all the bureaucratic channels, it was designated that Apalachicola Bay would be a high priority project, and that we could begin assigning assets and personnel to guide and understand the project to really start getting it going. And then this past summer, we had a stakeholders and logistics meeting down in East Point at the lab there, to really cement partnerships, understand what we're working with, understand the logistics and the best ways to make this work, because I've worked on coastal zone soil surveys from Massachusetts to Texas to California to Puerto Rico, and everywhere has different logistical challenges. So it's always a good idea to go out there and look at something before you start bringing, as we say, the circus with all our equipment down there. So what we determined at this stakeholder meeting was, based on the size, we really wanted to split this project into 3 phases, and each phase would take around 2 years. So phase 1 would really focus on areas around the Apalachicola River Delta proper, as well as a little further up the stream and in East Bay. We also decided to include Lake Wimico in this as well, to understand kind of the diversity in soils and what we're seeing around it. And then phase 2 would focus on the St. Vincent Sound, so everything pretty well west of the St. George Causeway. And so that would do an update inventory for St. Vincent National Wildlife Refuge, some of the oyster leases in the area, areas that are interested in it, as all the way down towards Indian Lagoon. And then finally, phase 3 would be the largest phase, and that would be everything east of St. George's Causeway all the way up to Alligator Harbor and passed up through Caravelle, to provide updates and inventories for that area. So when we were talking about in the initial conversation about what is soil survey, so of course, as much fun and as much as I would rather just go out there and start digging holes and getting to see what the soil is and how it differs, we have to do our pre-field work. And so that involves generating a landform map and kind of laying out plans of how do we, picking out what our landforms are, what are representative areas on those landforms that we're going to inventory the soil resources that we believe that it will give a good, clear understanding and look at what the soils look like in different areas of the bay, without going around and punching a hole or doing a soil description every 10 feet. And so we identified our landforms. In this example, I showed 3 of our landforms. So we have our mainland coves. So that's the uppermost one in the blue area. So these are generally areas that are in embayments, not quite a cove, where a cove is cut off with usually a single point of entry in our geomorphic description. But these are lower energy areas, fairly protected. And usually you see finer textured materials, more silty or clay than sandy. Our other landforms that we identify are our dredge channels. So for our purposes, we usually don't map areas that are more than 3 meters deep or 9 feet of water. And the reason for that is for our focus that these areas don't support vegetation. And we can get into the kelp forests on the west coast, but we'll leave that be for now. But that's why you see down here on the southern end of that map that you just kind of have this colorless area in between the barrier islands and the mainland, is because all that area, based on the bathymetry that we have, was deeper than 3 meters. So it's not what we map, and we'll leave it as a water soil map unit. And the dredge channels there, that's one of the second landforms that we were doing. So we identify that because, of course, that's been human altered, human manipulated. We don't want to map that the way we're going to map, say, like the lagoon bottom out here in the larger bay in itself. So understanding those differences and identifying all of these so that we have a good plan going forward when we put down our soil sampling sites, and that we know that we're covering everything we're going to see, and we can confidently and scientifically proper map all these areas. And so in Apalachicola Bay, in all 3 project phases, we actually identified 23 unique landforms. Usually what we do is for every unique landform, we gather about 5 observations at a minimum, and depending on the variation or variability of the soils that occur on those landforms, we might gather more observations. So staging and preparing equipment. So traditional soil survey, it could be 1 or 2 soil scientists, and they can go out with their soil loggers, they can do a soil description, stop in a cornfield, walk into a pine forest, do their description and walk out. Well, since we work in subaqueous soils as well as these wetland soils, we really need specialized equipment and research vessels to access them. So we kind of have our own fleet for different uses depending on what needs to get done where. But generally what we have is we've got a 26-foot research pontoon, and I'll show some photos of that in a moment. Our 24-foot research pontoon, which is this one you see here, you may have seen it out on the bay some last fall and winter when we were down there working. We also have a refrigerated trailer. So to keep this chemistry intact whenever we pull these soil cores, so you can kind of see those metal soil cores standing up on the stern of the research vessel right there on one of the pontoons. Whenever we pull these soil cores, because we're exposing them to oxygen or might not immediately be able to process them, we keep them in a refrigerated trailer. And so that refrigerated trailer is set anywhere from about 36 to 40 degrees Fahrenheit, and that keeps the chemistry from denaturing in our soil cores until we're able to get to them and describe them to make sure or get that done before there's any kind of degradation. And so we also use a lot of pipes. So what we use is 3-inch aluminum irrigation pipe that's cut into 24-foot sections. I'll show you a photo of that in a moment. And that's what we kind of use when we're taking these soil samples, because how it works is that we have a concrete vibracore engine. So what that is it is a generator that has a vibrator on the top of the pipe, and what it does is when you turn that engine on, it'll shake that pipe down into the ground, and we'll get it down to where we can collect about 6 feet of soil underneath the water column. And then just like you do if you were to put your thumb on top of a straw in a drink, and you pull out and you still got some of the drink in the straw, it's kind of the same idea with what we do. We put a suction cap on the top. We have our A-frame that we put up on our boat. We hoist the core out of the water, and then we put a cap on the bottom, make sure the rest of the top's filled up with water, so it's kind of keeping it ambient conditions. Cap it, tape it up, label it, and get it to the refrigerated trailer so we can do our description on that later. So here's some of the tools of the survey here. That top left photo is our 26-foot research pontoon. It's built like an absolute tank. It's a great boat. And you may have seen that as well out in East Bay and maybe parts of St. Vincent Sound last fall. That top middle photo where you see myself and my coworker standing in front of one of our refrigerated trailers, you can see they're pretty decent size. They're probably about 12 feet tall and about 18 feet long. So it's another piece of equipment that you got to acknowledge or think of when you're moving. And there's just some other photos of our vessels on the water. That's another one of the refrigerated trailers coming off from delivery on the right side of the slide as well. And then in the bottom left, that's a trailer load of our coring pipe that we'll use to extract these samples. So specialized training and a soil scientist. So just because we're soil scientists and we're out on the water doesn't mean we can just hop on a boat and go out there and do it. All of the folks that work with us get on our boats. They're required to pass the Department of Interior's Motorboat Operator Certification course, which that's a 24-hour course that has a classroom portion as well as a skills portion. And we also do self-rescue techniques right there. That photo in the middle is us teaching self-rescue techniques, floating techniques in the event that you get dislodged from a boat or some other issue happens. And the class really does a great job of teaching some of the environmental challenges as well as the navigational hazards and the rules of the road and how to properly operate a boat while you're out there, so we're not just letting folks loose to run out here and be on a boat. And of course, we get all that done. That makes for a good day on the water when we can do our job safely and accomplish what we need to do. So launching the project, we began field work in the fall of 2024, as I've mentioned before. So far, we've completed about 3 weeks of field work, and we've made very good progress. And in any given week, we have anywhere from 3 soil scientists to 9, depending on budgetary restrictions or folks' availability to get out there, to really get out there and get this work done. And right now, we were able to make great progress this past fall, and we're actually about a year ahead of schedule right now and hoping to keep it that way. So this is our sampling plan moving forward. So those red circles that you see on your map are places we've already sampled or we've already took soil samples from them. And so with all of these that are sampled here, we took cores at all of these locations that are red circles. The green squares are where we are going to sample. So that's future sampling sites. And these red circles, we described all these soils. We didn't necessarily take lab samples for all of them. Approximately half we took for lab samples that were representative, we believed, of our landform, landscape, soil survey concepts. And so approximately 73 have already been sampled. Green, we have about 81 slated to complete hopefully this fall and next year as well. And then we're really going to start moving into our terrestrial work, where we're going to work on inventorying some of the marshes, some of the tidal forests further up the Apalachicola River, as well as the marshes on St. Vincent and the Barrier Island, to give a proper soils update and understand what the soils look like and their characteristics are out there. So generating lab data. So these representative pedons that we do sample, what we're looking at is the particle size analysis, so sand, silt, and clay composition. You may have seen the USDA soil texture triangle before, so that's understanding where it falls on that triangle, which can dictate a lot of the interpretive uses for it. We're looking at the carbon and nitrogen content, so how rich or how carbon rich, how much organics are located in these soils. Bulk densities, so how compacted are they? Are they loose? Understanding that, and then oxidized pH, which is the presence of sulfidic materials. And so the importance of oxidized pH is essentially what we do over a 16-week period, is we have a wet and dry cycle of measuring the pH of a soil to simulate as if it had been dredged and placed on land. So some of you might be familiar with beneficial land use of dredge, where they take dredge from waterways or marinas and then place it on the land to help the tidal marsh maintain its elevation. And so the idea behind that oxidized pH is it lets us understand if that material is going to become highly acidic once it becomes exposed to the air over several weeks of being placed on a marsh or disposed of otherwise. And so the outcomes for our cooperators and our data users is we really want to help and make sure this data helps identify characteristics for oyster production and restoration. We understand that's very important to the economy and the culture of Apalachicola, so it's one of our primary focuses. Map the dominant soils based on our taxonomic system. I was mentioning to Anita earlier, I'd love to get more into the taxonomy of soil, but I understand that might not be everybody's forte. And then another outcome that we're really looking forward to is understanding the areas that are best for subaquatic vegetation restoration and maintenance, because we understand that's an interest as well. Okay, so it's enough about the plans. What does the soil look like? We understand it takes a lot to get there, get everything in order, get people doing what they need to do, but what does the fun stuff look like? What do the soils look like? So we're going to go into the initial results here. Like I said, I've got some good photos and I'll kind of explain what's going on with these soils, but I don't have all the lab data back I want. So we'll go through these here. So first, what you're looking at here is 3 soil profiles from Lake Wimico. So these are the 3 of those 4 red circles that are on that map of Lake Wimico on the left. And so the general characteristics of these soils are they are very fluid. And what I mean when I say they're very fluid is, if you take like a golf ball sized handful of it and squeeze it, it immediately runs out of your hand, kind of like if you were to squeeze axle grease, something to that degree. The environment's a lacustrine, so it's a lake. It's fairly contained other than the dredge channels going in and out the rivers. And so it's depositional. Usually any material that's put into the lake itself is not going to exit on an average day, and so it's just going to be deposited. So it's a very, very depositional environment. And one of the interesting things that we found in Lake Wimico is we actually found a submerged histosol. There I go, I'm using taxonomy. A submerged organic soil. So it's a soil that on average, the horizons within that soil have a greater than 12% organic carbon percent by weight. And so these are very, very carbon dense soils, and they're fairly rare to see in a subaquatic environment. So that was interesting that we found that in Lake Wimico, and I think that's the organic soil right there in the middle. And that one, I believe, is the red circle furthest to the left on that map. Something else to point out here is that first image of the soil profile on the left, down about 160 centimeters. These tapes are all in centimeters. That's what the numbering is there. You can see that there's a thin dark layer about 10 centimeters thick, and we think that was maybe an old buried surface, potentially during oceanic transgression, might have been the edge of an old marsh or a tidal forest at some point, but since with sea level rise and transgression, it became eroded, but it essentially got encased as more sediment was coming into the lake and depositing. But generally the soils in Lake Wimico are pretty homogeneous. Didn't see anything, 3 out of 4, of course the 4th was the organic soil, but generally pretty homogeneous. So in the sloughs and rivers, we kind of got this river and tidal interface that occurs in the Apalachicola River that makes it interesting to work in and see. It's much more higher energy. Material was based on the initial texture data that I got back. It's kind of a combination of sandy material and this more fine material, higher clay content. The Apalachicola Basin is known for having a large clay content coming out of it, but you get to see like the stratification. So if you look at that first soil profile on the left there, you can just see these bands going back and forth of different depositional periods, maybe of stability, maybe of hurricanes, high rainfall events, where you get more material moving up higher out of the watershed or maybe being pushed in because of storm surge. And so you get these different depositional events that are stacking on top of each other. And so kind of that darker horizons, usually it means high organics. Sometimes it can be monosulfides or highly reduced iron sulfate. And it can give off that color as well too in these environments. And so the grayness in these soils, including Lake Wimico and the ones moving forward, the grayness in these soils is because of the lack of oxidized iron. So generally what gives soils its red color, I'm from an area where there's a lot of high clay soils in the mountains in Piedmont of North Carolina. The soils that are red because they have a high iron content, high oxidized iron content. And so when that iron is put in a reducing environment, anoxic environment, that it changes colors and it turns gray. And so that's what the gray color departed on all of this is from. And so also in these sloughs and rivers, we saw some shell fragments, not a whole lot, a lot of plant detritus in there, dead plant matter from higher up in the watershed, or the shifting of the channels themselves based on the weather events going on. So in our mainland coves and bay bottoms, we talked about that a little bit earlier, that these mainland coves are, depositional environments, low energy. That water and that sediment that's being carried is coming out of these larger channels, going into lower energy systems and being deposited. And these soils had a much higher clay content than the other ones. So it's showing that all that more fine material, that clay and that silt, is settling out in these bay bottoms and lagoon bottoms and mainland coves. There's some soils that we saw that had a lot of oyster shell fragments in them, whether that be in situ or displaced during storm events. So these were the areas in East Bay, particularly these soils we're looking at here, that we started to see some oyster shell fragments in there. And so something that I had never seen before, and I had to ask some folks with more local knowledge, is you start seeing these red bands in a lot of these depositional soil cores. And so potentially one theory is that this was deposition from Hurricane Michael when the Apalachicola River watershed, essentially got flushed out during the storm event, that any kind of free sediment that was existing on riverbanks or higher up in the watershed just got flushed down that river, and you kind of get this red deposition. And we see this in a lot of our cores that are in these bay bottoms and in these mainland coves. So it's that was something that was really interesting to see in them. So we also took some soil cores on these dredge deposits. You can see they don't all, that one in the middle does to a degree exhibit this gray coloring. And these all have been human altered to some degree. It's been dredge material that's been placed back on the side on a shoal. You can see where we took those two soil cores there, right south of Apalachicola and the 98 bridge. And so much higher sand content. And generally you had some oxidation from the dredging, so that gives it that oranger color. So that iron is starting to be exposed because that soil was churned up. And it maybe at some point was aerated to a certain degree before being redeposited. And it did have significantly increased shell fragments. So the soil core in the middle, I think, came just east of the old St. George Causeway island that the bridge used to run across in the middle of the bay there. And then those other two came from those two red dots that were located there. So it's really, when you're doing dredge deposit shoals, you'll see anything and everything, just because depending on where the dredge came from, how actively was it churned up, how far did it go. So you really don't know what you're going to see on a dredge deposit shoal, because when you work in these environments enough, you got an idea that, in your mainland coves or your bay bottoms, these low energy areas, you're going to see more fine material. The higher energy areas, you're going to see sandier materials. But dredge deposits really are a wild card when it comes to that. And here we have the backside of the barrier islands. And as you might expect, there's quite a bit of sand on these. We did find some relict oyster beds, what it looked to be. So that soil core on the left, you can see at about 150 to 160 centimeters that we have a lot of shell fragments there. And so all we can say is that it was probably a relict oyster bed to some degree, maybe burrowing. And then during a storm event, it got encased. And so these are very high energy areas. These are what we call washover fans or washover fan flats. So when these barrier islands were forming and they were still smaller, there would be constant events during storm events where sand would get pushed over the barrier island itself and come out on the backside and kind of do a washover fan there. And so it's historic overwash, and these areas usually are very, very good for subaquatic vegetation. It's something we constantly observe in a lot of different environments that we work in, that these washover fans or the backside of these barrier islands where it's very high in sand content are good for subaquatic vegetation. So general observations and takeaways, kind of talking about what we've seen so far. Again, the stuff up near Lake Wimico, it's very fluid. You know, we got the surprises of our submerged and buried organic soils that were occurring up there. And it's highly acidic when oxidized over a period. So generally areas that get that are primarily freshwater and have freshwater river and input usually are more acidic when oxidized over time. So wouldn't use soils from Lake Wimico for dredge material, thin layer deposition. East Bay depositional areas are very high in clay. We see that stratification in the rivers and sloughs because of the changes in the deposition periods that are going on within them. And then in St. Vincent Sound, it's generally higher in sand. Deeper areas have increased clay, so as the water depth increases, as the water energy goes down, you get more of that clay deposition. But you know, there's areas on the backside of the barrier islands or right up against the shoreline where you get that constant shallow churning washing machine of the waves. It's pretty sandy. And the pHs of these soils were fairly high due to the marine influence, so there's not as much freshwater oxidation. You have those salts, sodiums coming in with that marine influence that kind of buffer your pH and keep it high. And there was some evidence of relict oyster reefs depending on where we were looking and where we were taking our soil cores at. So moving forward, the project was put on a hold for a little bit, but right now we got one or two more steps to go through, but we're hoping to continue field work this August. Hopefully the weather will cooperate and we can keep moving forward with this project. Right now, the phase 1 is slated for completion in 2027, along with phase 2, we believe. And then in phase 3, which is going back east of everything on the St. George Causeway to Alligator Harbor, that should be completed by 2028. And so the data will be published in Web Soil Survey and Soil Web, but we were talking about earlier our interfaces for the public to access these, and there are some of the links there as well. And so what will be available to you? So anybody that wants all the raw data that is developed from this soil survey is free to have it. You know, it's a taxpayer funded service. You know, you're inclined to have it as well, and we can provide it to you in Excel sheet. You can go through this portal and download it. But you know, it's everything from pH to the electrical conductivity, particle size distribution, organic carbon, the fluidity, so understanding like how you know, kind of the stability of the tidal marshes in these areas or the tidal forests that we've worked in, and also the associated interpretations. And, you know, this is always one of my plugs where it's like, you know, help us help you, because we want to develop soil data and develop soil interpretations that work for you, because our soil survey is not worth much to you if you can't get the information you need out of it. So if there's a certain interpretation that you need in there that you think would be valuable to you in a larger group, that's something that we can certainly work on and would love to partner with to make sure we get that information out there that you need and that we have done what we needed to do in this area. All right, so that's what I have. And you know, thank you for your time and attention coming out here and listening to what we've got going on with the Coastal Zone Soil Survey. There's my contact information right there, and I'd be happy to answer any questions that anybody has.
Okay, thank you so much, Reuben. Does anybody have a question? You can come off mute. You should be able to unmute yourself or raise your hand and I can call on you. I had a quick question while we're waiting for people to form their thoughts. Did you know, we had a conversation before we started about the project that the Riverkeeper is conducting over in East Bay about a lot of the sloughs and some of that material, for dredge material being washing down into East Bay. Do you have enough analysis to see something like that occurring, or over the last 50 years, is there a notable difference?
So how we look at it is we take soil profile descriptions down to about 6 feet or 2 meters. So if that deposition has occurred that recently, if we know what we're looking for in the respect of what that dredge deposit would look like on top of the natural soil, then absolutely we would be able to tell if it had been there. We did take 8 or 10 soil cores right out there on that delta area where the St. Mark's and Little St. Mark's drops out. And there was some sand deposition on top, not necessarily what we would consider significant, but you guys understand what you're working with and what would be concerning. And we are sharing that data with the river keepers and have sampled soils for them, surface soils as well, so they can conduct their analysis on those to hopefully better answer that question.
Great. I'm not seeing any hands raised. They may not be able to. They're all of our staff's in one room, so we'll give them a few more minutes.
Yeah, I mean, I'd be happy to talk about soil taxonomy too. I know that's a hot topic. Good. Anything else? I mean, happy to talk about any aspects of the project.
Yeah. Well, I think we're just excited it's getting conducted and we'll have the data. It's going to be a welcome addition.
Yeah, absolutely.
I'm not seeing, everybody's being closed mouth, I guess, today. I'm not seeing any hands up, or either we're having a malfunction in our question.
I know I didn't explain it that well.
Well, they might be absorbing it. And we do have your website and the ability to contact you. So I'm going to say that we might be having some technical difficulties on the questions front, but we really appreciate your time. And I look forward to maybe interviewing you again or having you on again in maybe 2026, talking more about the project. And I think it's going to be a wonderful addition to have. So, and we are recording this SciCafe, so it'll be available on our website, and we'll send links out to everybody. So thank you so much, Reuben. We appreciate it, and please stay in touch. And thank you all for attending.
All right.
And our next SciCafe will be August 28th. So thank you. I appreciate your time.
Absolutely. My pleasure.
Oh, wait a second. Wait. We do have one question. Oh, Megan commented from the research reserve staff. Great job.
Well, thank you very much. I appreciate all the assistance with the logistics they've helped provide with us as we work through all this.
Well, anytime. That's what we're here for. We love partnering with other scientists. So.
Absolutely.
Okay, thank you so much.
All right, thank you.
All right.
Y'all have a good evening.
Bye.